I think we're close. Good to go? All right. Good morning, everyone. Thank you for being here on a Monday morning at 9 o'clock. And it's really nice that you are joining us here today in the beautiful Apotex Center at the Bannatyne campus. Thank you as well for joining us online. Much appreciated. This is our sixth and final Emerging Researcher Spotlight series in the 2023-2024 season. My name is Rene and I'm the Associate Vice President Research. Let me begin by acknowledging that the University of Manitoba campuses are located on original lands of Anishinaabe, Inunuak, and Sinunuak, Dakota Oyate and Dene, and on the national homeland of the Red River Métis. As a member of the University of Manitoba community, I'm proud to be part of research networks that are in collaboration with Indigenous peoples, and by learning and relationship building, our research networks move forward in a spirit of reconciliation and collaboration. Today, we will hear from 9 presenters, and there will be a question and answer round in the end. The presenters are from the Faculty of Agricultural and Food Sciences, Desautels Faculty of Music, Faculty of Kinesiology and Recreation Management, Faculty of Social Work, and Rene Faculty of Health Sciences. And so today reiterates what we were hoping for to achieve in the Emerging Researcher Spotlight series. We aimed to achieve connecting people from different disciplines as presenters as well as participants. We know that such exchanges can lead to new research questions and collaborative approaches. The word cloud on your screen shows the topics that we will be learning about today. This word cloud shows the topics of all 44 presenters in the 2023-2024 Emerging Researcher Spotlight series. And I would like to thank Meaghan Abdelmasai for creating this word cloud and all of the other ones. Meaghan is a confidential assistant in the Vice President Research and International, and I would like to thank Meaghan so much for helping us to organize this Emerging Researcher Spotlight series. It was it's very hard work, but well done. So about 850 individuals to date engaged in the 2023-2024 Emerging Research Spotlight series. And I think this series has been well received, and I thank everyone for their participation. So it is now my pleasure to introduce the first speaker, and the first speaker is Jason Gibbs, who is joining us from the Faculty of Agricultural and Food Sciences. And Jason discovered a new species of bee in 2018, and the species is named after Dr. Gibbs, Apolus gibbsi. And if I mispronounce that, you can correct me, Jason. So Jason's research program focuses on integrative taxonomy of wild bees, molecular phylogenetics, pollinator ecology, and insect conservation. With that, please welcome Jason.
Thank you very much, Annemieke, for that nice introduction. Thank you, everyone, for coming. It's an honor to be asked to speak here with all these other great presenters. So mostly I'm just going to show you some pretty pictures of bees, because that's what I like. So as Annemieke said, most of my work is on wild bees. So if you're thinking honeybees, that's someone else in the department. So I work in the only department of entomology in all of Canada, which is another honor I'm happy to have. And as part of my job, I'm also the curator of the museum that we have in the department, the J.B. Wallace R.E. Ruffley Museum of Entomology, which is one of the largest insect collections in Western Canada. I always like to show this slide when I'm sort of in a broad audience. And in addition to the research I do, I'm also a teacher. So this is a quiz. And my question for you, and feel free to shout out answers, how many bees do you see on this image? I heard 5. Any other guesses? 11? Oh, someone's been doing their homework. That's really good. I don't know if my animation is going to work. So I see 2. That's the first one, up in the upper left, and in the bottom right. So there's all of the rest of those insects are flies. So even though probably in the last 10 years or so, you've probably heard a lot about the importance of bees as pollinators, and people are concerned about pollinator declines and the decline of bees. In fact, most of us are pretty poor at knowing what a bee actually is. And it's not a reflection on any of us. You know, it just takes time to learn. When I started my PhD, I didn't know what a bee was either. So on the upper part here, this is an onion flower. There's a honeybee, which you probably are familiar with. And the other bee is a sweat bee. And this is the group of bees that I did my PhD work on and sort of dominated my life for the last 20 years or so. And there's some similarities. They're both social. They both have queens and workers in their colonies, but they're obviously quite different in their sort of size and shape and the way they sort of live. The life histories of bees are very diverse and very complex, and part of the research, part of the fun of the research is getting to learn more about those. So this is just a few examples of bees that we have in Manitoba, and I thought I would just sort of show these to sort of contrast some of the different sort of life histories and behaviors that they have. The one in the upper middle is a miner bee. It's a solitary bee. She lives in the ground. She's solely responsible for provisioning her offspring. And they're very common. So if you look at any flower in Winnipeg around this time of year, you'll see this bee. And they're very likely nesting in front of your lawn. The one right below it, that red and yellow kind of waspy one, is what we call a cuckoo bee. It doesn't build a nest or collect pollen at all. She goes into the nests of the miner bee and lays her eggs on the provisions that she makes. And that's actually a fairly common behavior that's evolved multiple times across different lineages of bees. And so understanding those relationships and understanding how these different behaviors evolve is part of the research kind of work that we do. The two on the far right are both closely related bees in the genus Perdita, and they're specialists, and they're specialized on different flower groups. So the one the upper right is a specialist on composite flowers. It's on a gumweed, I think. And the one on the bottom right specializes on prairie clovers. And so it doesn't matter how many flowers you have in an area. It doesn't matter how good the nesting habitat is. If you don't have prairie clover, you're not going to have that bee. So they're very constrained in their sort of ecological interactions and the habitats that they need. And sort of understanding those sort of habitat requirements is also some of the work that we like to do. The one in the upper left is what we call a cellophane bee. So they actually line the nests of their cells with polyesters. So it's sort of a natural sort of material that they produce in their abdomens. And that's quite a rare bee. So this is it also specializes on prairie clovers, but only the silky prairie clover, it seems, which is an endangered plant in Canada. And so it's an exceedingly rare bee to find. And it's also lovely. It's a beautiful little bee. And the one on the bottom left is an oil bee. So it's also specializes on a particular plant, but only plants that have produced floral oils, which it incorporates into its cells and its provisions for its larvae. So there's lots of different habitat specializations, different sort of evolutionary sort of relationships amongst all these different kinds of bees. And sort of learning about that is what makes my work fun. As a taxonomist, you know, a big part of the work that I do is trying to understand kind of the who, the what, the where, the why, the when of bees. So are these one species or two species, and why is that, you know, why are they two species and not one, is kind of the work that sort of it dominates what we do. So if we want to understand pollinator conservation, it's important to know what species we have, what a species is, all these sorts of questions. And then where are they located? You know, why are they located there and not other places? when were they there and not in historical times and not currently, which is part of the value of the museum, because we have all these historical records to look at bee distributions and things like that. Yeah, so this is kind of the work that we do. From sort of an applied sense, we're trying to understand some of the drivers that sort of explain sort of bee diversity, whether or not it's land management, the effects of landscape sort of around a crop field or whatever it might be, climates, disease, whatever factors might affect bee diversity in an area. And then from there, try to understand sort of this ecological services the pollinators provide. So whether or not that it's in sort of a cropping system or sort of wildflower sort of pollination. And more recently, we've been sort of trying to incorporate some of the molecular phylogenetic data that we sometimes do. So that usually we use that to sort of understand the relationships of bees and try to understand the evolution of certain traits. But we're now sort of exploring the idea of using phylogenomic data to measure the amount of sort of phylogenetic history that's encompassed in a group of bees in a particular area. And this was from a paper that was published a few years ago that I was part of, where we were looking at the effect of land cover on bee diversity and the effect that that has on fruit pollination, in this case apples. And the number of species, the species richness, was not that much of a good explanatory variable. The abundance was a little bit better, but actually the phylogenetic diversity, like how many different kinds of bees and how much sort of evolutionary history that represents, was actually the best predictor of sort of the ecological benefits of those bees. So that is in a nutshell the sort of work that we're trying to do, and I will pass it back to Anamika.
Jason, that's great. Thank you for teaching us all about bees. Appreciate it. It is now my great pleasure to introduce Melody McKiver from the Desautels Faculty of Music. Melody's music integrates electronics with Western classical music to shape a new genre of Anishinaabe compositions. They are the 2020 recipient of the Canada Council's Robert Fleming Prize, awarded annually to an exceptionally talented young Canadian composer. Please welcome Melody.
Buju, Megwayanakwat and the go, Obishikokang, Mamatagoji, Watigwaning and Donji, Mashkade Bijake, Nadodem, and Shnabe, Nawe Nangwayabe and Dao, Bejik Made. Hi everyone, my English name is Melody McKiver. I am a member of Lac Seul First Nation, specifically the Frenchman's Head community within nearby Treaty 3, about 5 hours northeast of here. Thank you all for listening today. So in 5 minutes, I'm trying to integrate some of my research as well as some of my creative practice, what some of myself and my colleagues have been working under around a framework of which we refer to as Indigenous musical sovereignty. So on Ijitchikeek, what do I do? I'm an Indigenous scholar within the Faculty of Music, and so much of my work comes from being a working musician before returning to academia in the last couple of years, so both as a performer and composer. I specialize in electroacoustic works for the viola, and increasingly I've been working more in composition, both for chamber music as well as film and television. I especially enjoy working in composition for media because a really major current within contemporary Indigenous artistic practices is working within storytelling frameworks. As a politicized Indigenous person that writes instrumental works, it can often be hard to convey different discussions and meanings, and drawing in like larger social threads as an individual violist, but working collaboratively with different storytellers, different filmmakers, and really centering Indigenous artistic leadership throughout my various creative projects is something that's really exciting to me. So I've loved starting to work more on film composition over the last couple of years. Concurrently, my graduate studies were in Indigenous studies and ethnomusicology, and so I don't consider myself an ethnomusicologist by practice today, but by training. And so when I am working in a more academic capacity, I've been writing in a number of themes, whether it's music education, especially post-secondary, broad interest in Indigenous studies. I missed NASA this year, but I consider that more of my intellectual home, especially around Anishinaabe methodologies, Two-Spirit studies. Land-based practices are a big part of my non-musical creative practice that really continues to integrate my creative work as well. And yeah. And so for a music talk, I wanted to share a quick excerpt. So this is a music video that was shot with APTN a couple of years ago. It's called Debinoe Gijagon. An important recurring theme in my works is when I take the time to collaborate with different Anishinaabe elders within my community. So Debinoe Gijagon was a title that was gifted by Ralph Johnson, who's Anishinaabe knowledge keeper that lives and works out of Sioux Lookout, Ontario, which is within my traditional territories. And broadly, we were speaking about like the role of music that it has within traditional Anishinaabe spirituality, cosmologies, and that phrase roughly translates to sound is at the center of all creation. For the handful of music nerds that we have in the audience, you'll note that the excerpt is in a really strong 7/8 meter, where I was working with that sort of phrase, Debinoe Gijago, and trying to integrate some of that sort of rhythmic approach as a way of alluding to the Anishinaabe language. At the time, I was also working on a commission responding to Steve Reich, who's a Jewish American composer who also did a lot of work with oral history and excerpts and using kind of vocal speech rhythms as a way of deriving different melodic compositions. And it was a technique that I found really intriguing to derive for this particular work. And the music video was set within my homeland, so we see a bit of vistas of some scenes over Sioux Lookout and Lac Seul First Nation, a bit on our powwow grounds. Hopefully it works. Yes. So I had the clip timed, but it started at the beginning, so I'll just play it safe there. But coming back to the theme of Indigenous musical sovereignty, as a Anishinaabe artist that was classically trained in Western art music practices, specifically violin, viola. My mother's a pianist that I collaborate with regularly, who was also classically trained as part. Definitely part of this narrative and inquiry is like, why do we come to Western classical music as well? It's because my mother's a 60 scoop survivor. My late grandmother was a residential school survivor. So often thinking about the colonial and imperial dynamics of how Anishinaabe musical tradition has really been ruptured and interrupted quite forcefully and violently by my family. And so always negotiating where working with elders and different musical practices, but also recognizing that I'm giving this talk in English today, just as much as different musical systems have been integrated. So Indigenous musical sovereignty, we somewhat coined at the Banff Centre for the Arts. It's my colleague Chris Dirksen, who's a Cree Mennonite cellist who was raised in Edmonton, and similarly to me, Indigenous person, classically trained, plays strings. And within the Canadian sort of classical music community, like many society, like Canada-wide, there's been an increasing renewed interest in reconciliation, especially post-TRC. There's an increasing interest in having more Indigenous programming broadly across Canadian musical and where I think we are right now is kind of like Dances with Wolves equivalent in terms of the Canadian classical music, where like, yes, it's an Indigenous theme. There's a lot of buckskin and leather involved. It's a sort of story about Indigenous people, but the story is interpreted by Kevin Costner. So if we want to take Kevin Costner as the creative lead or the musical equivalent, that's a lot of what the handful but growing number of Indigenous people that are working in these different classical music areas are negotiating today, where a lot of the programming is still led by non-Indigenous people, even if they're telling Indigenous stories. So when we talk about Indigenous musical sovereignty, and definitely there's scholars, Trevor Reed out of the US is a Hopi scholar that's also writing extensively about sovereignty. Concurrently, I spent a stint working in public health and as a knowledge translator prior to returning to music academia. And there I was more studied in notions of ownership, control, access, and possession, which we see a lot in terms of data sovereignty, in terms of Indigenous research. That is a notion that I see less frequently in fine arts spaces. There's definitely a lot of creative liberties taken, but adjacent fields like Indigenous filmmaking, the Imaginative Film Festival always looks for Indigenous authorship. So even if it's Adam Beach has a role, it's not necessarily, it's Disney Squanto, it's not an Indigenous story. So that Kevin Costner piece as the director is really what I'm trying to encourage people to look at in terms of music as well. Like, is there Indigenous leadership or is there just Indigenous participation? And we even said, so ownership and control of our stories and artistic projects is of vital importance for Indigenous creators. The stories we need to tell at this time often significantly vary from the existing canon of Indigenous-inspired works. So there's been an explosion of these Indigenous-inspired works. Often they're quite re-traumatizing, really looking at I don't have the time quite to go into it, but I think this notion of Indigenous sovereignty also means that colleagues of mine like Andrew Balfour are really invested as well in sort of like Baroque choral music traditions, and not all of his stories necessarily are about residential schools, because as Indigenous peoples, we have breadth and complexity beyond single issues. And concurrently, and this is often our answer, because like you're Indigenous, but you play the viola, and a work is Indigenous when it's created by an Indigenous artist, regardless of theme or topic. A story is Indigenous whether it comes from ancestral knowledge, lived experience, or imagination. And much of, and this is work that was collectively authored by a number of colleagues that are both performers and composers across the Canadian classical music scene. We asked non-Indigenous composers that seek to tell these Indigenous-inspired works, be honest with yourself and ask why you feel compelled to tell the story and whether you are the right person to do so. And concurrently, access to resources is something that I think is really starting to encourage these sort of collaborative Indigenous projects within the arts scene, and many of my colleagues have experiences of people rushing in to write us in grants, often without our consent, to try and leverage Indigenous participation, even if it's coerced, as a way of accessing more arts funding. And so we are trying to communicate to the broader community that if these traumas are not your immediate familial stories, then perhaps you shouldn't be accessing like these Indigenous specific funding streams. And And so I have one upcoming chapter that'll be published next year in the Oxford Handbook of Orchestration Studies. So we called it Indigenous Voices and Ethics in Mixed Media Compositional Practice. And a lot of this framework of Indigenous musical sovereignty and pushing back against like mainstream Canadian classical music tendencies is informing that. But I wanted to flip the narrative a bit and engage with works of a number of different Indigenous artists that take that lead authorship role and artistic direction, and to further explore what it means to have Indigenous stories. And looking at like relationality, reciprocity, kinship as really important terms for a bit more of an ethical consideration of like, why is this person writing an opera about missing and murdered Indigenous women when like they don't have any missing and murdered Indigenous people within their families? It's how do you come to this story? How are you in relation? It's and also discussing with Indigenous artists when they do put up boundaries, when they say like, no, I don't want to touch this story. I will only do so in collaboration with this person. And in the photo is a friend and colleague of mine, Jeremy Dutcher, who's critically acclaimed for his work in a lot of rematriation of anthropological archival recordings that were taken from his community for close to a century and that he had to go to Ottawa to re-access. We were in the studio in Montreal there working on a forthcoming trap for my next album, and I also discussed Jeremy's work in my publication. Miigwech Gibbs and Dewalk Nungam. Thank you all for listening today.
Thank you very much, Melanie, and thank you for telling us about the great work you're doing. Up next is Dan Henhawk from the Faculty of Kinesiology and Recreation Management. And it's good to see you again, Dan, and thank you for being here. Dan's research focuses on multiple conceptualizations of leisure and the socio-cultural study of sport and recreation. In his work, he strives to privileged Indigenous ways of knowing and of being in the world. Dan, the floor is yours.
Great, thanks very much. Thanks very much for having me, Seiko. Good morning. My name is Dan Henhawk. I am an assistant professor in the Faculty of Kinesiology and Recreation Management, but I situate myself in sort of this broad field of leisure studies. Leisure studies is an interdisciplinary field that examines the phenomenon of leisure. Leisure for me is an umbrella term that encompasses a number of different areas. As examples, this could include the management of public and private recreation, outdoor recreation management, and or activities such as tourism or therapeutic recreation, and the sociological study of the various activities that we might deem as leisure. As a phenomenon, leisure is often thought of as those activities that we engage in. However, there are also philosophies of leisure that suggest that we can also conceptualize leisure in terms of how we think about time, for example, time free of obligations. In a sense, we could also think about leisure as freedom, like freedom to choose, freedom to be who we want to be. Additionally, we could also think about leisure in terms of the meanings that we create, thus giving ourselves a sense of identity and our lives a sense of purpose. For my part, I've always loved thinking about leisure and its role in society. However, being also embedded in Indigenous scholarship, I've also thought about the ways in which leisure, both the activities we engage in and the philosophies related to leisure, have been part of the colonization of Indigenous peoples and their lands. As such, I've spent a lot of time thinking about this relationship and the fundamental question of how we might decolonize leisure. Karen Fox, professor now retired from the University of Alberta, wrote that many languages, including many Indigenous languages, does not have a specific word for leisure like we do in English. This is not to say that Indigenous cultures did not have an understanding or their own words for leisure, but rather that an Indigenous conceptualization of leisure may differ from a Eurocentric perspective. If you take the Gunyagahaga language, my language, the Mohawk language, I don't speak my language, but as far as I understand, we did not have words for things like leisure. Something interesting about our language too was it was explained to me that it was very verb-based. So if you think about language as being verb-based, then how might that change how you see the world? So if you look at lacrosse as an example, one name or title given to lacrosse, its loose translation would be that it's called bumping hips. Also, in our language, maybe the closest thing that might come to thinking about leisure in terms of, living a good life or living the good life would be thinking about living with a good mind. Living with a good mind, though, requires you to understand the notion of Indigenous relationship and the relationality first. So understanding Indigenous perspective on leisure, however, requires an understanding of Indigenous ways of knowing and ways of being, which is encapsulated in Indigenous language. Since I grew up in a colonial education system which offered 1 hour of Mohawk language classes per week, and I didn't have a family who spoke our language, I currently can't speak my language. It's also difficult to re-engage with language when you live so far away from home. I'm originally from Six Nations, which is in Southern Ontario. However, with that said, this doesn't mean that we can't begin to understand an Indigenous notion of relationship, or what Sean Wilson refers to as relationality. This notion of relationship recognizes that entire systems of knowledge are already interpersonal, intrapersonal, environmental, and spiritual, including relationships with ideas. A particular note is Sean's statement that rather than viewing ourselves as in relationship with other people or things, we actually are the relationships that we hold and are part of. Indigenous knowledge is relationship because it develops through experiences that are already social, spiritual, and ecological, resulting in a communal understanding of the world, thinking of we first and being attentive and responsive to the people and those relationships in the world. So we think of a we first and not I, then again, how does that change your perspective on the world, and how does it change how you engage in the activities that we do? How might that change how we think about something as simple as leisure? As a result of this orientation to the world, embodiment of one's knowledge is highly valued, and it becomes evident in the way one acts, the things that they might make, and the ways that we shape our world. Fundamentally, then, this orientation to the world should ask us to think about what an Indigenous notional relationship means for how we currently conceptualize leisure, and also what this means for indigeneity and Indigenous identity. Sandy Grande, she's a Quechua scholar, currently living in the United States. I think she's still at Connecticut College. Wrote a book called Red Pedagogy. For me, it's a fundamental book in my studies in terms of thinking about colonization and education and Indigenous identity. She argues that we have, for a long time, been convinced that Indigenous people suffer from an inability to create a comfortable modern identity. However, by focusing on this seeming identity crisis, we ultimately obfuscate the real sources of struggle and oppression, which Sandy argues are colonialism and global capitalism. Indigenous identity is thus not, from her perspective, and I would argue from many Indigenous people's perspective, is not an existential crisis, although for many of us it is an existential crisis because we don't quite understand what has happened to us. But it is in actuality a crisis of power, the power to name, to shape, to control our lives, and to control our own labor. Thus, if leisure has been and continues to be part of the colonial project, both in the activities that we engage in and the philosophies that we understand it to be, then we need to decolonize it. We, meaning Indigenous peoples, need to rethink leisure from a position of relationship. To do this, particularly through research, is going to take engaging in research paradigms that also break the colonial cycle. This means that research that pushes back against the epistemic violence that has been wrought against Indigenous peoples and the disciplines that challenge dominant paradigms. For me, leisure studies is uniquely positioned to do so. This might include everything from narrative inquiry to storytelling, to various forms of creative analytic practice, to performance disciplines. As Denzin and Ivana Lincoln write, never have we had a greater need for a militant utopianism to help us imagine a world free of conflict, oppression, terror, and death. We need oppositional performance disciplines that will show us how to create radical utopian spaces within our public institutions. Thank you.
Thank you very much, Dan, for your presentation. And we're moving on, and it's my pleasure to introduce Rusty Souleymanov from the Faculty of Social Work. Thank you for being here, Rusty. It's good to see you again. Rusty's research examines how oppression affects the health and well-being of equity-deserving communities. He is committed to ensuring access to healthcare and social services for all. Rusty, feel free to start.
Thank you. Thank you very much. I work as an associate professor in the Faculty of Social Work, and in 2021, I've established this community-based health research lab called the Village Lab. And so I want to talk a little bit about that work. So as we know, health and social disparities disproportionately affect Canada's some of the most disenfranchised and marginalized and equity-seeking communities. So to address this injustice, our team at the University of Manitoba in the Faculty of Social Work became very committed to improving access to healthcare and social services for those communities. And so my interdisciplinary work spans multiple disciplines, including social work, public health, psychology, and social sciences. And the idea in my work, kind of my program of research, to better understand the relationship between socioeconomic marginalization and a variety of health outcomes. My particular focus is on the research in the realm of HIV, STBBI, which stands for sexually transmitted and bloodborne infections, harm reduction, broader social determinants of health, and all of that through integrated research, education, interventions, and policy. So in 2021, like I said, we've established this lab. And part of it was for a variety of very important reasons. For example, Manitoba is the second highest province in terms of HIV rates in the country after Saskatchewan. And we also have a variety of other issues. So all that work kind of made sense. It was timely. It was the right place to do this work. So we work with a variety of communities in our lab who represent different projects. All of this work is community engaged, community oriented, community based. So we work with Indigenous communities, Black African Caribbean communities, 2S LGBTQIA communities, immigrants and refugees, people who inject drugs, people who live with HIV. All of those are key and priority populations in HIV research and policy right now. So that's kind of the rationale for including all these different communities. What do we do at the Village Lab? Three main kind of overarching objectives. The first one is facilitating communities of practice. So we bring researchers, policymakers, important stakeholders to work on a variety of key issues affecting those key and priority populations in HIV. We're also strengthening capacity among emerging scholars, students, but also importantly, people living with HIV, people with lived experience in the community. So we're trying to strengthen their research capacity. So like I said, partners, peers, mentees, trainees, all work together in our lab. And finally, knowledge into action. So what's the purpose of research? It is to affect policies, affect social structures in which we live and find ourselves. So through our work, we want to influence policies, services, practices, while also prioritizing accessible knowledge sharing with our communities. A variety of values drive our work in the lab, including EDI, decolonization, social justice, inclusion, and human rights. This is our wonderful team that's doing all the great work, with our knowledge holder and Elder Albert McLeod on the right, who is leading and guiding most of our work and is also a knowledge holder in the lab. So there's a variety of projects that are currently going in the lab. Some are more active projects than others. I'll kind of go through some of them just to give you a little bit of idea of what these different types of work that we do and what is kind of my program of research. So the Gigi Bapamin study, which translates from Ojibwe We Survived, is a project that looks at the health and well-being of Indigenous people living with HIV in Manitoba and Saskatchewan. Like I told you, the two provinces mostly overrepresented in HIV cases. So we've uncovered quite a lot of interesting data, looked at how, for example, the COVID-19 pandemic affected substance use for these populations and their access to healthcare, uncovered quite a lot of anti-Indigenous racism in healthcare and how all of that affects communities' access to different types of social and healthcare. Ubuntu Pamoja study is a project that focused on HIV self-testing among Black African Caribbean communities. So the project is still ongoing, and we've uncovered a variety of testing methods that are most appropriate, acceptable to communities who are part from the ACB community. Migration and health study focused on creating migrant-affirming care in Manitoba for a variety of refugees and migrants who are living here. Manitoba to SGBQ and men's health study focused on healthcare access for two-spirit, gay, bi, queer men, with a particular focus on how homophobia and transphobia influences people's access to care in our province. The HIV doula project. This is our new project that focused on extending the work in reproductive health and mixing it with sexual health. So doulas are individuals who provide a variety of support for women in particular in terms of birth work, but also death work. And so we try to reconceptualize HIV as a life transition and work through the model of doulas to really kind of see how can doulas be trained in HIV prevention and then can deliver the HIV prevention work right at the time when it's needed. And then the other two projects, one looked at the impacts of COVID-19 on the community, and Investigators is our capacity training program that we deliver in the lab specifically for 2S LGBTQIA communities. So just to give you a few kind of pictures from our work, that is to say that all the work is very community oriented. So we have community guiding circles that really tell us how to do the work, what are some of the key priorities in the community. So the research idea never originates in the lab, it actually originates in the community and then comes to this kind of the research lab. So this is our work in Saskatchewan and Manitoba. This is the community guiding circle for the Ubuntu Pamoja project. So community really gets the priority to decide what's the research objective is. They actually themselves conduct research. We analyze research together, then we send them to conferences and other events for knowledge exchange and dissemination. So we really try to build that capacity in the communities through a variety of means. As a matter of fact, we hire people with lived experience, we train them, we provide networking opportunities for knowledge exchange and translation. So this is kind of a little gist about kind of my community-based research in health. Thank you so much.
Thank you very much, Rusty. Thank you for telling us about the great work you do. Our next speaker is Jennifer Yamamoto from the Max Rady College of Medicine. Jennifer is also affiliated with CRIM, which stands for Children's Hospital Research Institute of Manitoba. Jennifer's research focuses on improving pregnancy outcomes for women with diabetes and their babies. Thank you very much, Jennifer, for taking the time to inform us about your work. The floor is yours.
Perfect. So thank you so much. Hard acts to follow with diabetes, but here we go. So my research program focuses on how we can use technology to improve outcomes in people living with diabetes and pregnancy. And so we'll start with diabetes. I think most people here know what diabetes is. Diabetes is either when your pancreas can't make enough insulin or the insulin you make doesn't work. And that's important because insulin allows you to use sugar, and then you get hyperglycemia. And there's a number of complications due to hyperglycemia. So 11 million Canadians right now are living with diabetes, so really common, about 1 in 10, and 1 in 3 live with prediabetes or diabetes, and 1 in 10 pregnancies are complicated by diabetes now. And the reason why this is really important is because we know that diabetes increases the risk of a number of pregnancy complications. Now, hyperglycemia is very dangerous for a developing fetus. And so what do we see if we see really high blood sugars during pregnancy? Well, a lot of adverse outcomes. So things like babies being very large. This is a big concern of my patients. Low blood sugars in babies, so needing IV dextrose or sugar to bring blood sugars up at birth. Being admitted to the neonatal intensive care unit, which is a very costly, and very traumatic experience for many parents, and preterm birth, so babies coming a bit too early, either because we get them to come too early or because they come too early naturally. Now, these are all common, but less severe. Unfortunately, pre-existing diabetes is still, to this day, despite all of our advances in medicine and diabetes care, associated with an increased risk of severe adverse outcomes. So these are things like birth defects, so heart and spine problems, as well as neonatal death. So these are very serious complications, and we're seeing these about 4 to 6 times the rate we would in people without any diabetes. And so there's good news and bad news. So the good news is that we know that helping people to get really tight blood sugars both before they get pregnant and during pregnancy can decrease the risk of a number of, well, all of the pregnancy complications I mentioned. However, getting these really tight blood sugars is exceptionally difficult for people living with diabetes. We can really only use 2 medications in pregnancy, insulin and metformin, and this is exceptionally challenging. People living with diabetes who are pregnant work exceptionally hard to try to reach these targets. And so really what my research program is aimed on is, can diabetes technology bridge the gap between what we see as our current diabetes and pregnancy outcomes and what we see as optimal diabetes and pregnancy outcomes? And how are we doing that? So 5 minutes, not a lot of time, but I'll talk to you today about one of the technologies that we do use, which is something called continuous glucose monitors, or CGMs. Now that it's summer, you can probably start seeing them on the back of people's arms. And so this is about the size of a quarter. It sits on the back of the arm, and some sit on the abdomen, and it checks sugars every 5 minutes. So it gives you a lot of detailed information. So this is kind of what it looks like. So most of the sensor sits just right on top of the skin. And you can see that there's just a tiny little filament, a tiny little sensor that sits in the fluid underneath the skin. And what it does is it senses glucose underneath the skin every 5 minutes and then communicates via Bluetooth to either a smartphone, an insulin pump, or a reader. And with measurements every 5 minutes, we get about 288 measurements per day. And the alternative is finger poking. So nobody can finger poke 288 times a day. And so these have really revolutionized diabetes care. So really, I don't have much time to talk about these, but a couple of my studies are seeing how we can use CGM to improve outcomes in people with diabetes. So the first one is predisposed. So in this one, we're actually putting on glucose monitors after people deliver with gestational diabetes to see if that can predict diabetes better than our current very sugary drink test, which anyone in here who's taken it knows it's terrible, and almost nobody likes it. And then the other one is something called Concept Kids. So this is in people with type 1 diabetes who use continuous glucose monitoring in their pregnancies. We're actually looking at their kids to see if those continuous glucose monitoring metrics can predict things like executive function, ADHD, autism, which we know are a bit more common with diabetes exposure in pregnancy. So with that, I'll thank my amazing team, my colleagues, collaborators, funders, patients, and participants, and for you for your attention.
Thank you so very much, Jennifer. And now we are joined by Meaghan Jones, also from the Max Rady College of Medicine. Like Jennifer, Meaghan is also affiliated with the Children's Hospital Research Institute of Manitoba. Meaghan's research program focuses on environmental exposures during prenatal and early childhood periods and seeks to establish when and how these become biologically embedded. Meaghan, feel free to start.
Thank you very much for the introduction. Like Jen said, this was a hard act to follow. What an amazing group of people and a really amazing variety of things we're learning about today. I'm not gonna lie, after bees, Indigenous music sovereignty, I don't know if everybody's gonna love learning about epigenetics, even though it's my favorite thing, so I'll do my best. So my research really looks at this one fundamental question. How do early life environmental exposures, things that happen in utero or in the early childhood period, go on to influence health later on? It's a concept that we kind of understand, right? It's something that we get sort of viscerally, but we don't really know how it works. So this is really founded in this idea of the developmental origins of health and disease hypothesis. That's a very specific health theory, sometimes called the Barker hypothesis, named after its originator, David Barker. And the idea is, in the early life period, we have these sensitive sort of windows of opportunity where environmental exposures can have long-lasting outcomes. And there is a lot of evidence for this. There are things like nutrition, socioeconomic status, inhaled pollutants, which is my main area of research, are linked later in life to all kinds of things. Heart disease, diabetes, mental health, asthma, and COPD are really common ones. And again, that's what I really focus on. The early evidence from this comes from studies talking about in epidemiological studies. So you look at a group of adults. One of the classic ones is the Framingham heart disease study. This was done out of the United States. And they were looking at heart disease. Was sort of men who were medical doctors in the 50s. So these men were all fairly affluent, they all had similar jobs, but they had really wildly different rates of heart disease. And then when they sort of went and said, what was the number one determining factor about when these men developed heart disease, it was birth weight. And it was both ends of birth weight. So either you were born really small or born quite big, you would have had an increased risk of heart disease later in life. But this is decades, 50, 60, 70 years between that sort of early life environment, which is we think birth weight is a proxy for, and the heart disease. So these things exist, these connections exist, but how does it work? So I think that epigenetics is one of the ways that this works. So we can think about epigenetics like a form of cellular memory. It's how cells remember things. These are a set of chemical marks. They work with DNA. They turn genes on. They turn genes off. The number one role of the epigenome, the first thing that it does, is it helps cells remember what kind of cells they are. So different types of cells have different types of epigenetic marks. But they also help cells remember past environments. And we can see this in a short scale. If we take cells in a dish, we expose them to something, they turn some genes on, they turn some genes off. When that goes away, some of those little chemical marks stick around. So we are trying to expand that idea into sort of a broader sort of life course study, try to see whether epigenetic marks actually are left behind by early life exposures and are linked to health outcomes later on. As you can imagine, it's not simple. So at my lab, we have three main areas of research, three main sort of approaches we take to trying to study this. We do population epigenetic studies. So these are typically large-scale data sets that we get we get epigenetic marks from hundreds of people. We have hundreds of thousands of sites across the genome that we look at. We have these are often really well-characterized birth cohorts, so we often get samples from babies or from children, where we know a lot about where they lived, what their air pollution exposure where it was, what their diet was, things like that. And that helps us answer questions like, what kinds of early life environmental exposures are leaving behind these marks? When does that happen? So we like this idea of windows of opportunity. How much is required to leave behind these marks? And we're starting to get into modifying factors. I mentioned diet specifically. So I study air pollution and diet, and we think that actually a high quality diet reduces the impact of air pollution on these epigenetic marks. But that only gets us so far, because human population studies, while super powerful in that way, it's hard to get at really specific mechanisms. So we're getting into animal and cellular models. That'll help us answer questions like which cells are involved. I mentioned that each cell has a different epigenome. When you do human studies, you're limited to what kind of cells you can get. When you're doing cells in a dish, you can culture any kind of cell you want. That gives it more at the developmental context. Mice grow faster than people, and so we can sample more frequently. We can understand that whole life course in a year instead of having to wait 70 years, which can be quite challenging. And it really gets us this idea of timing. We can actually use controlled exposures to understand what windows of opportunity are the most sensitive in terms of these environmental exposures. The third aspect is sort of our forward-looking and our aspirational part of this, which is the translation and intervention section. So we're starting to understand how these marks work, which environmental exposures are doing things, but so what? What can we do about it? Obviously, reducing air pollution, stopping smoking, those things are great, but those don't help people who have already been exposed. So what can we do about it? So that's the idea of moving into these translations and interventions, trying to link the epigenetic marks and changes we're seeing specifically to health outcomes, and using these to test whether interventions are reducing those signatures that are left behind. So this is an ECR, sort of emerging researcher spotlight, and I like to say, when you start a lab, when you start as an early career academic, people say, pick a lane, right? Do your thing, pick the thing that you're really good at and do it really well. I did not do that. I did a lot of things. I consider myself a bit of a scientific generalist. So in these sort of three areas, we've got a lot of projects going on. I also have a fabulous team of people that does this work with me. These are all my students and staff that have worked with me over the last couple of years. So in the population epigenetics, we've got a number of cohorts work going on. The iCare and NextGen cohorts are Manitoba-based cohorts. They are looking at similar to Jen, we're looking at exposure to diabetes in utero, what kinds of health outcomes that has. And we're similar to what Jen is saying, we're looking at the actual molecular epigenetic marks in there. The CANDLE study in Memphis, Tennessee is air pollution and asthma. Moving into the animal models, we've got a mouse model of early life exposure to cigarette and cannabis smoke. The AirSafe project is our new huge CFI-funded project that's going in across the street, where we're going to be able to expose mice and cells to air pollution. We'll be able to generate our own specific, we know exactly what's in it. We'll be able to use that to expose cells and get controlled exposures there. We're getting into epigenetic editing. So if we see these marks induced by an environmental exposure, what happens? Can we get rid of them? Can we actually go in and remove them? And will that get rid of this outcome? So we're getting into this, we're starting to move into that sort of translational intervention stage. We've done some anti-aging and cancer therapeutics and looking at epigenetics of those kinds of things in treatments. And then the future work is we want to get back and bring this back into the population sphere again, right? This is why this whole thing is a big circle. And of course, I couldn't do any of this work without all the fabulous people that work with me, our research collaborators, all of the different birth cohorts and the participants and families. So thanks, everybody. That's my work for you.
Thank you very much, Meaghan, and thanks for reminding us about AirSafe that was funded by CFI-IF. That's fantastic. Up next is Rene Zahedi from the Max Rady College of Medicine. Rene's research primarily focuses on understanding the causes of diseases using mass spectrometry to study changes in proteins related to diseases. Although newer to the University of Manitoba, Rene is a full professor and has published more than 140 peer-reviewed articles, including in Cell, Nature Biotechnology, and Nature Communications. Thank you, Rene, for being here and being part of the symposium series, and the floor is yours.
Yeah, thank you for the introduction and for the opportunity to present here. So, first of all, I realized my title was maybe too much focused on health research, because that's what I usually do. So I'm maybe not going to heal the world, but I'm going to try to heal disease. So what we're doing is a technology called protein mass spectrometry that's very fantastic. And why are we interested in proteins, and why is it going to change the world? So we heard already from Meaghan very well that there is definitely a difference between genome and what comes out of an organism at some point. The genome is our blueprint, but epigenetics, many different environmental factors will affect what's ultimately going to happen. That can affect identical twins, but I also have this nice example here of a caterpillar that turns into a butterfly that are genetically identical, but of course very different in what we call their phenotype. So we have a difference between the gene, the genotype, and what we call the phenotype, which is what the organism actually is at that time point and what is actually happening. So what happens is the genome encodes for messenger RNA that you all know from the vaccines, that will turn into proteins. Proteins can be, for instance, insulin, enzymes, antibodies. There's thousands of different proteins in every cell of our body. And they actually are the main drivers that regulate all the biological processes in our body. And that's also why most of the drugs, of the Food and Drug Agency that are approved actually target proteins, because these are the guys that also make us sick. And why also most of the biomarkers that we use to detect disease or to determine which treatment is good are also proteins. So what is mass spectrometry? It's a very interesting technology. The instruments are pretty expensive, but what they basically do in a nutshell, they allow us to weigh molecules or molecule ions, very accurate. So even if we had two piles of sand at around 70 kilograms, we could differentiate them based on a couple of milligrams, which we call is an accuracy of 3 parts per million. And we can do that very sensitive. If we take sugar, my God, I forgot the word, sugar, whatever. Put it into a volume of water that corresponds to 2,500 Olympic pools, and dilute it basically and analyze it, we can still detect it. So it's very accurate and very sensitive. And we can use it now to study those proteins, these thousands of proteins in different cells and how they differ. Because we have thousands of different proteins that are not present in the same concentration. Some proteins are there in many different copies, some are in a few, and they can change between healthy and cancer, for instance, in this case. So we're trying to understand between different diseases how these patterns change across thousands of proteins. That allows us to analyze biomolecular mechanisms, fundamental mechanisms that control cells and tissues, identify, for instance, novel drug targets for novel diseases where we don't know how to treat them, or, for instance, we also want to identify what we call biomarkers that allow us early detection, which we hopefully all know is always very important to get a successful treatment, or also to basically understand which patient will benefit from which kind of treatment. We're generating tons of data, big data, so we're using more and more artificial intelligence to understand this kind of data. And ultimately, we want to turn this into clinical assays. We can use very tiny amounts of blood or needle biopsies, which are something that, for instance, in a cancer biopsy, see it's very small. This is a millimeter. This is approximately 1 microliter of blood. So very small amounts of samples we can use for patients and try to find those markers, for instance. We've been using it for a variety of different things. For instance, develop new methodologies that allow us to do new kinds of studies. We also have been using it for understanding some more fundamental things in biology. Or here's an example where we use this technology to understand why certain cancer patients don't respond to the drug as well as they should. People never understood why, and this is something you can, for instance, find out with this kind of technology. So, this one I also wanted to mention. This is where we actually analyze dental enamel from mummies of the medieval ages to actually determine their sex, because all other different technologies didn't work. So I think we can try to heal the world with this heal disease with this kind of technology. And there's a lot of labs around the world are doing big progress on this. And why does it take so long? Because we want to do it well. You may remember this lady here. She was 2014 on the cover of Forbes. She was promising she had it tested with a tiny drop of blood will allow you to detect hundreds of diseases. Turned out, as all scientists would be able to tell you at that time, that it was all fraud. So she's now in prison, and we don't want to end up like this. We want to do it well. That's why we're working together in collaboration in the Manitoba Center of Proteome Systems Biology between technologists, research clinicians, and disease mechanism people to really do this well and together in collaboration. This is my acknowledgement slide, and thank you again for your attention. Thank you.
Thank you very much, Rene. And yes, I remember that story coming out about the fraud. So our next presenter is Cedric Tremblay, also from the Max Rady College of Medicine. Cedric's research focusing on deciphering molecular mechanisms underlying blood disorders and developing innovative therapeutic approaches. Like Cedric, he's also available. Cedric is also affiliated with CRIM, as well as the Paul Abrechtston Research Institute, Cancer Care Manitoba. Please welcome Cedric.
Thank you so much for the kind introduction. I have big shoes to fill after all those amazing talks, so I'll try to be brief and clear, and I'll put my timer to make sure I don't go over time. Here we go. Sorry. Okay. So the main focus of my lab and pretty much my career for the past 20 years is I've tried to understand diseases that affect kids. Since I started my postdoc in 2008 in Montreal, I mostly focus on acute lymphoblastic leukemia. And so here's just a graph showing that even though cancer affects every population in the country and in the world, the type of cancer that patients develop depending on age is very different. So here on the far left, you can see that leukemia is the main cancer in kids, so 0 to 14 of age, whereas brain cancer is actually the cancer that kills most kids in Canada. And that distribution strongly varies when you look at adolescents and young adults. And so you have leukemia here, which is 1 in the 10th more common cancer, but it's the second cause of death in adolescents and young adults with cancer. And we still don't understand why kids and adolescents and young adults respond differently to treatment as compared to adults. And so that's what's been the main focus of my lab. So I've been focusing on leukemia. So leukemia is a type of blood cancer that you find in the bone marrow. It affects white blood cells, and it's widely distributed, but mostly very common in developed countries. You have 2 types of leukemia. You have chronic disease that arises more in elderly people, and you have acute leukemia, which is most common in kids, and it's the disease that progresses very fast, so we have to act quickly to make sure that we can treat the patients. And so the main challenge, as I mentioned, is acute leukemia is very fast, so it's very aggressive disease. And so when kids or adolescents and adults get diagnosed, the leukemia is already full-blown, so you have invasion of leukemic cells you can find in the blood, you can find in the bone marrow, you can find in different organs. And what we don't really understand is what's happening before that. And I'll come back to that. Then patients get treated with chemotherapy. Some of them respond, some of them don't respond. We still don't understand why. And then they undergo, some of them will undergo remission, so that's called minimal residual disease, when the threshold is below the detection limit, so you cannot actually find the disease anymore. And in some cases, and in the case of pediatric leukemia, most cases will relapse, and so the disease comes back. And what people have been doing for the past 30 years is they take blood samples and bone marrow samples at diagnosis, and then they look at the relapse samples and they compare both, and they try to imply from that what happened. The issue is some patients, when they relapse, it's been 20 years or 15 years since their first diagnosis. As we know, we're not in an isolated bubble. We're exposed to different things, life changes, and of course it's very hard to comprehend what the disease come from and how it evolved when you're considering all those exterior factors that could have affected the disease. And so to overcome that, what we do is we use two strategies. The first one was use genetic models. So we have mouse models that recapitulate somehow the disease. And so by using those models, we can actually track the cells from early on, the pre-leukemic stage, and then track the disease progression, mimic chemotherapy treatment by treating those animals, looking right after chemo to see how the tumor has evolved, and then looking at the relapse. And we can compare those different states and try to understand how the disease evolved. The other way we can do that is using avatars. So we take patient samples, for example, we put them into immunocompromised animals, and we make patients into a mouse. And by doing so, we can actually treat them with different drugs and make preclinical trials in the animals, and we can mimic the patient. The advantage of that is we cannot keep poking patients everywhere. So the animals, even though it's done in an ethical context, we can actually get many samples and we can track the disease. And the other limitations that there is with leukemia is we still don't know where the disease originates. And so we cannot look in the patient to find the cell of origin, whereas we can do that in the animal. And so that allows us to literally track the disease and track its progression. So thank you.
Thank you very much, Cedric. And now, our final presenter of today and of the whole series in 2022-24, no pressure there, Filiz Koksel from the Faculty of Agricultural and Food Sciences. Filiz is the 2021 recipient of the Terry G. Falconer Memorial RH Institute Foundation Emerging Researcher Award in the Applied Sciences category. Filiz earned this award in recognition of her accomplishments in bringing Canadian crops from the field to our labs and in transforming these crops into foods with superb palatability and nutritional value. Filiz's research focuses on processing of plant-based foods, including plant-based meats and dairy alternatives. Filiz, the floor is yours.
Thank you very much, Annemieke. Thank you very much, everyone. It's a pleasure to be here presenting in this session. I'm an associate professor at the Food and Human Nutritional Sciences Department, Faculty of Agricultural and Food Sciences. And my research program focuses on food processing for a sustainable and secure food future. And maybe putting that topic more into today's context. The world population is growing very rapidly, and we have been hearing more and more about concerns related to food security and food sustainability. And in order to feed the growing world population, we need nutritious foods with lower water and carbon footprint. And with that in mind, an emerging food market is plant-based ingredients, plant-based foods, and these type of foods include anything from bread and baked goods, fermented foods, breakfast cereals, puff snacks, vegan, vegetarian patties, fake meat. And maybe my work is on the plant-based ingredients and food side, but also flexitarian options so that we blend these food ingredients or foods with animal-based foods so that we can reduce our environmental impact a little bit more. To process these foods, my research aims to better understand the unit process operations that go into transforming these ingredients into healthy, nutritious, and appealing foods. These unit process operations are operations that are being used very frequently in the food manufacturing environment, and they involve things like mixing, milling, for example, from grains to flour. wet and dry fractionation to fractionate the starch-rich and protein-rich fractions from flours to make ingredients like these, for example, protein shakes that you see everyone shaking at the gyms. Fermented food, so fermentation, baking, and extrusion cooking. So today, I do not have time to go over all of these unit process operations, but I'm just going to focus on extrusion cooking. I don't know if any one of you is familiar with extrusion cooking or extruded foods. I guess the most popular extruded food is Cheetos. Extruded foods include foods like puff snacks like Cheetos, breakfast cereals like Cheerios. More recently, we hear more and more about foods such as texturized vegetable proteins that can partially or fully replace animal meat in products like taco fillings or vegetarian burgers or fake meat chunks. So all of these types of foods can be processed using this technology. You can use several different types of ingredients. Here on this slide, you can see mostly the protein-rich ingredients. Those are the ones that are mainly used for making plant-based meat alternatives. The process itself is simple, but it brings together several unit operations that I already mentioned. So we start feeding the materials in a barrel under pressure and high temperatures. We have screw elements that mix that material. We add water, make it dough-like. We increase the temperature, we cook it, and if it's a puffed product at the end, we use different dyes. So it could be like a Cheetos-like product or a Cheerios-like product, but the work that I recently focus on is making plant-based meat alternatives, so creating these structures, these textures from globular plant proteins into like these fibrous layered structure that could mimic the mouthfeel of animal products, but without the environmental impact as much as animal proteins do. In this slide, I'm showing a wide variety of ingredients, the technology that's being used, and then different types of products that we can produce using that technology. So there is like all of these ingredients, the process, the end product quality, all interact, but the question is how they interact to affect that end product quality. And that end product quality could be physical quality, so that texture of that vegetarian burger when you bite into it, is it as juicy, or if you're biting into it like maybe like a puff snack, is it crispy enough, crunchy enough when you add more protein or more dietary fiber into it? Or it could be nutritional quality. For example, does it have the same protein digestibility, or does it increase your glycemic index? Like, does it have a high glycemic index when you consume these types of foods? So the overarching goal of my research program is to understand how processing food quality and the structural and mechanical properties of food ingredients interact. So, for example, if we test the mechanical properties of the ingredients or the intermediate products that make up a food, from that information, can we identify the end product quality? Can we identify these at earlier stages of the manufacturing process so that we can estimate the product quality before a food is fully manufactured? And to answer these questions, we employ techniques such as thermal imaging or X-ray microtomography or low-intensity ultrasonics, techniques that are maybe more commonly used in health sciences or techniques that are more commonly used in material sciences, and apply these techniques to food materials so that we can better understand the properties of these complex materials. And this way, I hope that my program will be able to ensure that we put nutritious, appealing food options onto our plates with better understanding of these of the properties of these complex materials. And with that, I pass it on back to Annemieke.
Thank you very much, Filiz. And if the last talk make you hungry, we do have some snacks outside there, so feel free to help yourself. We're going to now go to the question and answer round, hopefully answers. And just wonder if someone wants to start us off here in the room or online. We also monitor questions online. And we have a question there. Jackie.
Sure, thanks, Annemieke. So thanks to all the presenters today. I had a question for Meaghan Jones. I was wondering about the population-based studies about air pollution. I was thinking about how you might differentiate, because I would think that exposure to air pollution would also cluster with a lot of other things like maybe soil pollution, heavy metals, lead exposure, asbestos exposure. I'm thinking about my family who grew up in Weston here, where there were stories about, you know, having to close the playgrounds because the soil was so contaminated. Oh, excuse me. So that's sort of my first question, like how are you able to differentiate that? And then you also referred to the effects of a high quality diet being maybe a mitigation factor against air pollution exposure. And I'd just love to hear a little bit more about that.
Oh, Meaghan, I will get you the microphone so people online. I don't know if you mind sitting over there. That might be the easiest for the microphones. Thanks.
Excellent questions. So the metrics we use for air pollution, we have what we call land use regression models. So they are reported. We've got sort of sampling stations that are situated around a city. Most of these are from urban environments. So we're not dealing with agricultural waste most of the time in terms of soil pollution. But we use these land use regression models that geographers that are much smarter than me model nitrogen dioxide and different types of particulate matter. And so they measure it throughout a city. They use topography, wind patterns. It is a super complex model to assess, and we do it by postal code, both in Canadian studies and in American studies. We do it by postal code, so that's the sort of resolution we get on the air pollution measurements. We, most of the time, do not have soil pollution. Sometimes we have in-house air pollution monitoring. So in one of the studies we work on, the child study, it's a Canadian cohort study, we have a subset of kids that also had essentially like a sticky trap in the house, where we were able to, we took the dust from the home and used mass spec, where'd Rene go, to figure out sort of the in-home pollutions as well. And we found overall that they're fairly highly correlated. So yes, there's definitely other things going on there, but we are specifically modeling the air pollutants, and we model them separately. So that's the other thing about this, is we actually see independent effects of NO2, PM2.5, and PM10. Those particulate matters, the numbers mean the size. So smaller particles get deeper into the lungs. We think they have a worse effect in that way because some of them actually can sometimes get into circulation. But the bigger particles, they stick up in the airways higher, but because they're bigger, they have more stuff stuck to them. So they bring along more things with them. So we think that they have independent effects. So that's now I'm realizing quite a long answer to that question. But then the other thing you asked about was the diet. So this is a study, we just finished it. We've got the paper written, we're gonna submit it soon. So what we saw was, this was in the Candle study in Memphis, Tennessee. So we saw a high quality maternal diet as measured by it's called the Alternative Healthy Eating Index in Pregnancy. We saw a global reduction of the impact of air pollution on epigenetic marks in these kids if the mom had, and it was a linear relationship, higher quality diet, less effect of air pollution on epigenetic marks. The negative control we had was red meat. And it had the opposite, more red meat, worse effects. So that's a cool study, so keep your eyes peeled. That one's gonna come out soon.
Thank you very much. Speaking of meat, Filiz, this is something I've always wondered about. Why do they make plant-based food look like meat?
Good question. And not all plant-based alternatives to meat look and taste like meat. The more traditional ones that could replace meat include things like seitan. That's, I don't know if anyone is familiar, so made from wheat gluten, or tempeh, a fermented food made from soybeans, or tofu. It doesn't have that fibrous layered structure, right? So there are alternatives. But I feel that this the way of thinking about, the plant-based meat alternatives should taste and, have the same mouthfeel as meat is, I feel like it's more of a North American thing. I guess having seen, like when people want to replace meat with other products, I think they're, like, food is, in our cultures, right? It's, every day. So we're not only looking for, like, we're looking for the whole experience. So if it's like a barbecue, then people are looking for an alternative that will bleed like a beef burger when it's on the grill, right? So, but I think it's more of a like a smaller population that really thinks that it has to have the exact same experience. I mean, there's innovation in the food industry that could make that happen. But I also, but making those foods, these types of foods aren't easy. They use lots of ingredients that are really purified, and that kind of, works against this whole sustainability idea. So my research focuses on how we can make foods that can maybe partially or fully replace animal products, but made from the whole grains as much as we can, rather than extracting the proteins from this, proteins from that, mixing it that, and make it into a fake product. So there's so much that cereals, pulses, oil seeds can offer without that experience of animal meat, and that's what my research focuses on.
Thank you so much. I appreciate that. I'm looking at the audience, whether there are any questions in the audience or online. I see, question from the panelists.
Yeah, I have a question to Meaghan, actually. So I guess these epigenetic marks are probably irreversible, right? So yeah, so you can avoid them by changing the diet. But do you think you can also one day, find a way how to set artificial epigenetic marks to counteract the bad ones you already have accumulated?
Yeah, I mean, I'll tell you in 10 years. That's what we're working on. So because the studies that we do, especially in the human population structure, it's hard to tell whether we're preventing or reversing, right? We're taking the samples that exist, right? These are people that are happy, healthy kids walking around. So it's hard to say what's going on there. In the cells, in the mouse models, that's what we're actually trying to see. What do we do can actually to reverse those epigenetic marks? Because you're right, they can be. We think that sometimes they last, and that's why we have this long-term health impacts from early life exposures. But the goal is, can we reverse them?
I have to watch you. Thank you very much for that answer. Jennifer, I have a question for you. When I was driving to this event, actually, on CBC radio, they were talking about diabetes, and they were mentioning that it has increased over the years. And just wonder if you can tell us why that might be, and they were also expecting that it might increase into the future as well.
Yeah, diabetes has increased substantially over the last few decades, and all of our projections continue to show it going up. Like most things in medicine and health, the reason for that is multifactorial. So there are certainly some genetic components. We know there are some environmental exposures, for example, type 1 diabetes, which is autoimmune diabetes. There is also an environmental exposure that triggers the insulin deficiency. And then it's also a disease caused by racism, caused by colonialism, caused by stress, trauma. Like we're learning more and more that those are really important indicators of diabetes, and maybe something we used to say, oh, that's the genetic, predisposition. I think we're realizing that that's not true. Part of it is obesity, part of it is stress, part of it is diet, exercise. So it's a very, very complex condition that I would, we're always looking for a cure. We're always trying to get rid of it. I'd love to be out of a job. Going up. And so really important to find ways to treat people with diabetes in a more holistic way, take away some of the kind of burden of having diabetes, so make it easier on people. Certainly work on the stigma surrounding diabetes, which we know is substantial. So lots of work to be done, but definitely more and more common.
Thank you so much, Jennifer. I have a question from the audience.
Hey, this is a question for Rusty. There he is. I was just really interested in the idea of the HIV doula, and wondered if you could just tell us a little bit more about that.
Yeah, absolutely. So this particular project started with kind of understanding our numbers in Manitoba and Saskatchewan in terms of HIV. So right now, the way that the context looks like is the highest numbers are specifically among Indigenous populations and women in particular. And the reasons is similar to some of the other reasons that our colleagues mentioned: colonialism, anti-Indigenous racism in healthcare. People are less likely to be treated. People are less likely to be diagnosed. Our care is not women-centric in particular, so a plethora of intersecting issues is why the rates of HIV are higher among those populations. And so we were trying to find different ways to address this. And so one of the ways is we decided to work with our Indigenous partners and organizations. So we've partnered with Ganiganichik, an organization here in Manitoba. And the idea of kind of working on the doula project is to extend, to connect the reproductive health and sexual health. And right now we're doing research to understand better what are some of the needs in the community, how, if we were to train people in the community in terms of HIV doulas, how will it look like, which organizations will have the capacity to actually train, funding to provide this type of work. So right now we're trying to understand what the position will look like. Then later with Ganiganichik, we're going to go to the provincial government and start funding those positions, right? At Ganiganichik, but also other organizations, Indigenous organizations, mainstream organizations. So that's really kind of what has emerged in terms of this idea. So our first phase is to understand what is needed to support this position. Our next stage in the project will be to actually train people. So we'll provide training, and then people will go in their communities and do this work. So that's kind of the basic idea behind this.
I had another question. Thanks, Anamika. This one is for Jason. I thought that was such a cool presentation. I have a son who's completely bug crazy, so I'll have to see if I can bring him to visit the museum sometime. You mentioned using the museum to study historical populations of bees, and I assume other insects as well. I'm just kind of wondering about, like, how well does that work? Or are there a lot of gaps? I kind of maybe have a bad image in my mind of like a gentleman scientist sort of, collecting things that he's particularly interested in, but, of the past, but not maybe doing a systematic survey or collecting, more broadly. So, I mean, does that work well for you? I'm just curious.
You hit the nail on the head as the challenge. So usually we have to try to bin the data because someone will collect, Norman Criddle was an entomologist who was collecting around 1914 until 1930s. So we have a lot of data from him, some of his colleagues, but then there'll be a gap of 40 years where just no one's collecting bees for that period. And so if you want to look at a particular group of insects and see if the population's changed, then you have to gather what you can and try to pool it into a reasonable sample size. And then you're sort of measuring it that way. And then trying to look for, and then you end up with very few data points in your trend line. So I was part of a study that looked at that in the northeastern United States several years ago. And we basically ended up with 10 data points that were looking at this patterns of declines across different groups of bees. And in the initial analyses, we did it and we had one line just for bumblebees. And it was there was no change. But we knew there was a change because we had seen from basically the year 2000 onward, certain species of bumblebees just dropped off. So we had to sort of think about that differently and analyze the data differently to sort of catch that pattern because it was missed in the broader line. So you're right, it's tricky.
Thank you very much. I have a question for Dan. So we are increasingly leading busy lives. And I just wonder if you think about leisure, how many hours a week is actually really good for us? And why aren't we doing enough of it?
How many hours? Good question. One of the things that I've pondered over the years is this idea that we have to hit a minimum number of hours for, say, exercise or time away from work or something like that. And I don't think that there's any answer to that question, really, because part of that question revolves around how do we define leisure. If something is intrinsically motivating and we enjoy it and it's something that we do by freedom of choice. So if you're studying your bees and that's what you do for your work, is that also your leisure?
Yes.
Right? So I think one of the things is to recognize that within our lives we need to find different avenues to maybe de-stress or to change our focus, to give ourselves the freedom to be able to look after ourselves. And I don't know whether you can put a number on that. It's just a matter of trying to figure out, what works for you, because it's complex. Life is complex, right? And I think the more we try to compartmentalize it into numbers of hours or numbers of days, then that becomes also problematic too.
Thank you so much. Question over there. I'm going to do a little walk here.
Thank you. It's a question for Melody. During your presentation, right before your song, you kind of pointed out the significance of or you mentioned the 7/8 time signature as the music nerd in the audience. I was curious if you can expand a little bit on the significance of it and why it was used in that context.
Sure. So it was related to the phrase that Ralph Johnson shared with me, "Debin awe gijigo." So da 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 da. So it's just exactly like a 7/8 pacing, where I was thinking like a bit of the melodic contour too, where he goes kind of "Debin awe gijigo." So there's a bit of that up and down lilting as he speaks through Anishinaabe Moin. So I was really just working with the rhythmic meter of a syllable count based on the words, and then using that as a bit of kind of the underlying ostinato that I was playing on the piano.
Thank you very much. See another question on that end.
This question is for Filiz. I'm wondering if culturally, because in North America, we're all about the meat and our diets are more based on meat, and you see all of the advertising with all the burger companies and stuff now that they're saying plant-based. Is it the same idea, let's say in Europe or across the world? Are they so specific on doing this mimicking of meat with plant-based products, since there are a lot of cultures that just adopt those plant-based diets anyways?
I think that's mostly in North America, a little bit in Europe too, I think. But I think Asia in general is doing very well because they have these, traditional foods that are plant-based, yet still very rich in proteins, maybe made from like whole grains. So instead of just extracting the proteins from plant sources, they're getting the whole grain and all the nutritional components that come with that. I mean, there's going to be trends, right? So it's everywhere around the world. But I think it became like a very sexy topic during COVID, and maybe around that time, it's kind of, I feel like it's slowly going down a little bit because consumers are becoming more and more health conscious, and when you look at the nutritional properties of these products that are meant to mimic the animal meat experience, to make them have those properties, you're adding lots of sodium, for example. So there's a lot, like the ingredient list is, so large that, it goes a lot into making that product resemble an animal meat experience. So there are trade-offs. If you're really after that taste, then yeah. But at the same time, if you're looking at it from a nutritional perspective, maybe try and find other options that are healthier alternatives, and there are healthier alternatives to that. And I feel that Asia is doing much better in that sense compared to North America, because they have all these traditional and fermented foods that are readily available in the market.
Thank you very much. Question here.
My question also is actually for Filiz. I noticed on your slide that a lot of the sort of, we'll just say, fake meat products are, and I've noticed this in the grocery store too, are made with things like wheat and barley. And as a person, and it's becoming more common, I'll out myself, I have celiac disease, so I can't eat wheat or barley or rye. And a lot of these products, not all of them, there's become more, but particularly a lot of the fake meat products are made with gluten. And I was just wondering, I mean, I can kind of see, because if you're gluten-free, I don't know if I'm the loner in the audience, you can eat something really bad that's gluten-free and that falls apart. So I can see how using gluten-based items to make these meats would help them adhere together better, and stuff like that. But I'm just curious if you think that maybe it will start to become more common to see more gluten-free things. Like, of course, tofu is gluten-free, but you mentioned like seitan too, like there's certain things I can't even eat that, right? And I'm an omnivore, but I don't mind eating animal-free products or vegetarian products. It's just harder for me to buy them. Do you think we'll start to see more leaning towards gluten-free? Say, it doesn't have to be fake meat. It can just be like vegetarian or vegan products. Mm-hmm. Do you think that will come eventually down the line?
It is tough to be a celiac in this world. Because gluten is everywhere, and it's such a techno-functional protein that none of the other breads that we make using other grains closely resembles bread made with wheat, right? And its techno-functionality is so useful in the food industry that it goes into all of these foods that you do not expect. Right. And it is one of the go-to proteins in the meat alternatives industry with soy. And those two are the two technofunctional proteins that are easier to texturize and that are easier to mimic that animal protein experience. But there's been more and more research on alternative plant proteins. For example, pea protein is one of the biggest ones, and big companies like I'm not sure which one, the Impossible Burger or the Beyond, they have like a fully pea protein based fake or alternative, I should say, meat alternatives that are solely from pea proteins, among other ingredients in that ingredient list. But as the main protein they use, because there's more and more people searching for alternative proteins in this food sector, not because maybe they're celiac, but there's this all, concerns about consuming gluten, even if you're not a celiac patient. And there's lots of allergenicity issues around wheat and soy, right? So I think there's more and more research coming out. And pea is one of the stars now, but I've been working with hemp proteins as an example, faba bean proteins. And I was thinking when I put that slide on with all of those ingredients, it just looked like a list of ingredients that could be used, but their techno-functionality is so different and the products that they make are so different. And I think it's on us as the food scientists, food engineers, food technologists to understand these complex proteins a little bit better, their techno-functionality a little bit better, so that we'll be able to develop those appealing, healthy food products for all sorts of tastes and needs.
Thank you very much. Unfortunately, I have to end it here. So we have still have 15 minutes for some nibbles and coffee and tea and mingling with the panelists. So I would like to thank the panelists. One more round of applause. This was wonderful. And so this concludes the Emerging Researchers Spotlight series. And you will be able to find the entire series on the YouTube network. So feel free to check that out. And speaking of which, I would like to thank the entire IST team for all of their hard work to produce these excellent events and making it able to put it online. Thank you all for being here today on a Monday morning. It was a great event, I think, and it was a great honor to being part of this and listening to the groundbreaking work of the researchers at the University of Manitoba. We will be back in the next year. Thank you, Ekosi, Miigwech, and merci. Thank you.